what we call experimental questions. When you talk about experimental questions, here we are talking about investigative questions, which you cannot go to the paper without knowing it. What are some of the things you need to know? And how do you answer this paper or investigative question? There are some things we don't miss when it comes to the investigative question. You want it or not, it will be there. Let me start with what you call the independent variable. So when you talk about the independent variable, I've been telling you that if, for example, the table is like this, the first column, in most cases, the first column is always the independent, and the second column, it can be second or third, depending on the question, it becomes the dependent variable. So when you look at that, what does it mean? When you look at that, it means that this question use what call ID, which means that independent means dependent. So whatever you find in the first column is independent. Whatever you find in the second and the third, we call them dependent variables. Sometimes the question can come with more than one variables, like more than one dependent variable. So it means that you have to know that it can happen like that. However, sometimes when we bring this, we don't bring the table, and we bring a statement which tries to show you that or is talking about that experiment which is they have done. Sometimes if you get confused, there are some things which we use to show you that this is independent, this is dependent. How? A question can come and they say that when you see the word on, with, yes, for example, the effect of birth rate on the population of South Africa. The effect, so the statement before on becomes the independent. It becomes the independent. The question after, sorry, the statement after becomes the dependent. The effect of birth rates on the population of South Africa. It means that how is this birth rate affect the number of individuals in South Africa? That's what it means. So the effect of this on this, then it means that that's the meaning. You understand? The effect of this on this, then that's what it means. So it means that whenever you see on, yes, it means that the first is independent, the second one is dependent. I think I'm clear there. If you are not getting it, let me know in the comment. Sometimes we even use with. How does this vary with? So in most cases, what we talk about first is the independent. What we talk next is the dependent. I think that one is very clear. Ndivuo, are you there? I hope you understand what I'm saying. Let's go to the next point on what we call the experimental questions. Once you identify the independent variable, the dependent variable, sometimes we ask you about the reliability and validity. How do I identify and how do you write this reliability and validity in exam? Because it is not just that you just memorize the way they said it in exam. or in class, and then you end up writing it that way. No. Basically, reliability. How do I increase the reliability of the experiment? Number one, you can talk about the increase of sample size. But now, you don't just say increase of sample size. It must depend on the question they have given you. They can say that in this experiment, they did an experiment and then they used 200 people. The question can come and say that, how did the investigator increased the sample size? No, increased the reliability. So now, when you're answering this, it must be based on that. How did he increase? They're not asking you, how do you increase the reliability? How did this investigator increase the what? The reliability. It means that here you have to talk about that he used, you see, in past tense, he used 200 people. He used 200 people. How do you increase the reliability? So now, by increasing the number of people in the investigation, or more than 200 people, you see. So it means that now, yes, in other words, this is the sample size they are talking about. Unless they have not talked about anything in there, then when you can say by increase in the sample size. Books, most of the books, they will say that increase the sample size, but when you're marking, we might not give you the tick. Why? Because it is a cram, it is not an application. You have to apply based on the question they have given you. So you increase the what? The sample size. I think I'm clear there. Don't write this increase sample size. First read the question and see which sample size did they use. If they are asking you in a past tense, you have to answer in a past tense. If they ask you in present tense, you have to answer in present tense. Another question is, how do we increase the validity? Or how do I know that this is valid? The experiment is valid. So here, when you talk about validity, you have to talk about something which is supposed to be repeated. Oh, that's not only a reliability point. When you repeat the experiment, you're increasing reliability. You repeat the experiment and you get the average. Sometimes you repeat the experiment, or you get the average of what you have repeated. You understand? So sometimes if they ask you, how did he increase the experiment? How did he increase the reliability? You say that he repeated the experiment 10 times. So if the number of times they have used is shown in the paper, please show it even in your what? In your answer. He repeated the experiment 10 times. So that's how you can increase the what? The reliability. All right. How do I increase the validity? How do I increase the validity? Or how do I make sure that the experiment is valid? So that's the meaning. So here, you have to use what used in the first experiment. You must use it even in the second experiment. For example, use the same environmental conditions. But in this case, you don't just say same environmental conditions. No. You use environmental conditions, for example, same temperature, same light conditions. You expose to this experiment, or you carry the experiment based on the number of hours. So if experiment 1 of the first trial is 10 hours, the second trial must be 10 hours, the third trial must be 10 hours. And if so, it means that you have to use the same time. If you are using a female of age 10 to 20, you have to keep that in mind, that even the second experiment or the second trial, it must be 10 to 20. Even the third, so that is, you are increasing the reliability. Kundai, I think this is clear. Kundai, let me know that this is you understanding what I'm trying to say. Another question. Just a moment. Some technical issues here. Yes, I'm back. Ne? I'm back. Sorry, guys. There is something which happened with the technology here. All right. So, yes, I'm back. Ne? It was just a technical know-how, a technical issue. Ne? Yes. All right. We are saying that uh, how how we are talking about how do you increase the reliability. I think that is done, guys. There is no paper which is going to come, and in this paper you have not been given this question concerning about experiment. There is no way. So go to the paper. When you know that, you're gonna find that in your paper. I think that is done. um, Sometimes they can ask you the control experiment. Control experiment means that why do we use control experiment? Basically, we do control experiment to bring about the comparison, to compare the results, because control experiment is being done in normal environment, normal conditions, without being used in any external stress or what. You understand? So control experiment is just basically in a normal condition, things act like this. But in experimental, because you want to do a factor, then it means that things must happen like this. Yes. uh Then let's go to the next question, which is about um, graphical representation. Let me do like this. Yes, graph. I'm not going to waste too much time here. I'm just going to show you that sometimes when we bring one, either is a pie chart, a line graph, or a bar graph, and to know which one are you going to use, or either a pie chart or a bar graph or a line graph, you have, for example, sets of results. I said it like this. For example, there are two, and then here you have countries. Countries may be SA, USA, and then Canada, whatever. Yes, and then here you have maybe growth rate, growth rate, and then maybe here you have 20%, you have 30%, then you have 50%. You see, that's how it is. So once you see this can't be a line graph. This can't be a line graph, because the line graph, it must have 2 points, this point and this point. How are you going to plot this Canada, SA, here? here. So it won't be, where is SA here? So it means that you have to know that this is not a line graph. This one can either be a pie chart or a bar graph. A bar graph. I think that is clear. Then number 2, now let me go into detail. When they bring a pie chart, yes, if they bring a pie chart, what does it mean? You don't use these percentages, no, because a pie chart cannot be in percentages. It cannot be in mass. It cannot be. The circle, I say that the circle is 360. 360. So what does it mean? What does it mean? It means that you have to add this. When you add this, for example, 20 plus 30 plus 50. You understand? The good thing, this is already in percentage. So it's already, you know, it's out of 100. But if it is not percentage, it's something else, grams, the number of people, you add it and then you get the total, which is 100, depending on the question. So now, to convert this into degrees, now you're going to say SA is equal to 20, it is 20, divided by the total, which is 100, times 360. And then now I get the answer here. You understand? For example, if I check here on my my tool, it's not giving me. Yeah, on my tool, it's gonna be 20, yes, divided by 100, yes, times 360. Answer is 72. So it means that this is going to be 72 degrees. So when I'm plotting, I'm supposed to plot what? 72. So when I'm using my protractor, remember protractor is like this, ne? It's like this. So I'm supposed to bring this protractor, I put it here, and then I measure anti-clockwise 72. If 72 is here, then I put 72 here. You understand? And then now I go to another one. I have to calculate for 30. Maybe 30 divided by 100 times 360 times 360. I also get the answer. You understand? Now I put the protractor like this now. And then I also measure. You don't go back here. No, you don't go back. I measure. So the remaining one must be for 50%. And then I also convert it. And then if I convert it, it's going to give me 180. So that's how you're supposed to do the pie chart. I think I'm clear there. Then when you go to the bar graph, here on the pie chart, don't forget to put a key, don't forget to put a title, and then you put the key and then tell us what with this shaded. Don't write too much in the pie chart. No, don't write too much. Yes. So when you go to the bar graph, I say that when it's bar graph, please take note of this. First of all, you need to know the title. It must have the two variables, the one for X and the one for Y. The variables you're gonna write here. It must have these variables in the caption. Yes. Number 2, when you go here, look. When you go here, when you start plotting, for example, USA, whatever, maybe Canada, whatever, not Canada, is okay. It must be straight line, eh? Yeah, it must be a straight line. Once you have this, what does it mean? Now, after that, how do we mark? This bar must be equal to this bar in terms of width. It must be equal to this bar in terms of width. This space must be equal to this space. You understand? If there is another one, it must be equal to that space. Please take note of that. Put a title. Here, it must include units. Units. Here, no units. No units. Even here, it must include units. But countries. So that one is simple. And then now, when you have this, here, if you look at this, you're seeing that 20%, 30%, 50%. You see a person is writing here 20%, 20%, 30%, 50%. This is gonna be wrong. Why? Because this is the range is not the same. From here to here is 20. From here to here is 10. From here to here is 20. So it's supposed to be the same. 20, 40, 60, 80. You see, the range is the same. Or 5, 10, 15, 20, 25, 30. I see. It must be the same. I think I'm clear there. Eh? If you are not understanding, please let me know. Let me know. Let me know so that I can smash it. I can smash it, guys. These techniques, any paper you're going to write, come back and watch this video, and you'll tell me after the paper. You'll tell me that if you really benefited or you didn't benefit. And obviously, the answer is going to be what? It's going to be you benefited. Yes. Nkensani, do you understand what I'm saying? Yes. If you have any question, let me know. Nkensani, I'm waiting for you. All right. Then, when we go to plotting scale, I've talked about the scale. Line graph is also the same. Line graph is also the same. It must have figures. Units include, put units. Title, no units. Here, listen. How do I know that my line graph is wrong? How do I know? I will only know if I have such line graph. We don't have this. This is not met. If you have this kind of graph, it means that you swapped the axes. That's what it means. You swapped the axes. So a line graph, it might either be like this or like this or like this, but it can't go this way, it can't go that way. You understand? Going like this is fine and going down is fine, but this way it cannot happen. It is not acceptable in that regard. So the next question is supposed to be the scale. I talked about it. Plotting. When you are marking, we have letters we use. So I'm not going to talk about too much of the marking because we are now want to kill the paper tomorrow or the other day, whatever you're gonna write it, or today or whatever. Yes. The last question, I'm just talking about now the calculation before I go to the real topics. But this, what I'm talking about, you will find questions which are concerning about that. Percentage decrease and percentage increase. We also ask you that percentage increase and percentage decrease. These are some of the calculations I'm talking about in life science. Yes. When talk about percentage increase, yes, please repeat, how do I plot a pie chart? Okay, Dimpo, then can you please repeat the pie chart? What do you mean by should plot it anti-clockwise? I want it, okay, this one saying that it means that some people they didn't understand what we call the anti-clockwise or the clockwise. All right, let me show you what I'm saying, what I was saying before I go to the percentage increase, percentage decrease, just in 2 minutes. This is what I'm saying. This is what I'm saying. This is the protractor, ne? And then this is the anti-clockwise. This is the clockwise. So it means that it's a clockwise, this is anti-clockwise. Let me check if I can find here a protractor, and then I can show you exactly with the what, with the photo. Yes. I can't find one, but I think let me just continue here so that you understand better. So this is anti-clockwise. This is clockwise. Ne? So now, look. After calculating, for example, you said 20 out of 100 times 360, it gives you 70, whatever. I'm just giving a figure. Ne? 72, whatever. 50 divided by 100 times 360, it gives me 180. Ne? And then we are saying that we are saying 30 divided by 100 times 360. I'm just giving you and then you get any figure. Now you draw a circle using a compass, and then now when you draw a circle using a compass, you bring your protractor. Remember, it has scale from 0. Look at your protractor. You'll see what I'm saying. Look at it. Yes, it has a scale 0. It goes like this, 20, 180. It goes like that. Yes. And then now it has another scale which comes from here, 0, and then it goes like that, goes like that, and then this is 90. Yes. And then 180 is this side. So now this is the scale I'm talking about. Ne? Okay. When you put a protractor here, ne? Now the protractor is gonna be like this. It has two scales. Are you seeing? It has two scales. The scale from this side to this side and scale from this side to this side. So now if I measure from here and then I say I'm measuring 70. So I'm gonna use the scale which goes this side. So I measure, I say 70 is there. Because I'm moving in anti-clockwise way, that's why I'm saying that you measure anti-clockwise. Then you find it 72 is there. So you make it maybe XX, you come here and say key, XXX means SA. You understand? Then now, you bring the protractor now here. You see, the protractor is going to be like this. It's going to be like this. It has scale from here, going this side, and this had a scale going this side. Still use the anti-clockwise. Ne? You can use this the clockwise as long as, but if you use clockwise, you have to use clockwise throughout. Use anti-clockwise, you have to use anti-clockwise throughout. So now you put it here, then you measure 180. 180 comes until it reaches here. You understand? Then you shade it maybe Y, and then you come here, you say Y means maybe USA, you see, or Canada, whatever. And then now you put now the protractor here, you see, it must now face. So you turn your paper and then it faces that way, you see. So now it means that when you turn your paper, it's gonna be like this. You turn the paper and then now remember, you stopped here, you see. You put the protractor again, and you measure until you finish. That's what I'm trying to say, using clockwise and using the anti-clockwise. I think Dimple and Kensani, Nkanyisa or Nkensani, yes, I think I'm clear there. If I'm not clear, let me know. Lastly is percentage decrease, percentage increase. When we talk about percentage increase, percentage decrease, what does it mean? It means that the formula is always the same. Final, you have to find the final, you have to find the initial minus initial divided by initial. You see? Times, because it's percentage times 100. You cannot go to the paper because we like these things. But now, how do I know? For example, they want to know the percentage growth from 2020, 2000 to 2020. You understand? Yeah, maybe the growth here was 10 and the growth here was 45. So final, which is 45, minus initial, which is 10, divided by initial, which is 10, times 100. You have to show the working because we mark the working. Always we mark the working. So you get the answer here. I think I'm clear there. Then, if it is you started with the, for example, 50, and then you end up with the 30, still you're gonna say 30 minus 50 divided by 50. The answer you get here is gonna be in negative. It means that is a percentage decrease. While here the answer is gonna be in positive, which means percentage increase. Nkensani, I'm still waiting for your response, and Dimple. Yes. Then, when you go to, yes, I'm answering the questions people asked me yesterday, and then because I talked about them, but some people, they didn't understand them better. So another one is the last calculation is base calculation. Base calculations. Yes. When you talk about base calculation, remember adenine goes with thymine and guanine goes with the cytosine. Now, if this is left, this is right. Ne? If you have 10 left legs, how many right legs do you have? You have 10, because these things are compatible. So if adenine is compatible, or it's not compatible, is complementary to to adenine is complementary to thymine, and guanine is complementary to cytosine. If this is 30, obviously this is 30. So it's 30 plus 30 gives me 60. You understand? Okay, Dimple, I see your answer. I'm waiting for Nkensani. Yes. So if it is percentage, so it means that it's gonna be now 100 minus 60, and then I get 100 because it was percentage, it was 30%, minus 60, which means gives me 40. Yes. So if it gives me 40%, what does it mean? If it gives me 40%, what does it mean? It means that this 40% is for 2. So it is going to be 40 divided by 2, which gives me 20. So it means that it's going to be 20, it's going to be 20, and then 30, 30. So that if I add them, it must give me 100%. 30 plus 30 is 60, plus 20 is 80, plus 20 is 100. So it means that my answer is correct. So sometimes we bring them in percentage, but sometimes we don't bring it in percentage. What do we bring? We bring it in, we bring it in numbers. For example, if the DNA has 2000 bases, ne? Only 500 is guanine. What is the percentage, or how many bases are for the remaining nitrogenous bases, or the ratio? They can ask any question they would like to ask, but the answer remains the same. I'm seeing Mpiwe saying, I don't understand. You have to specify what you don't understand. So we are saying that here, if the total number of bases is this, and then only 50% is guanine. So I have to come back there and I say that guanine, you start with what is given. Guanine goes with the cytosine. So if guanine is 500, automatically cytosine is also 500. So I'm going to get 500 plus 500, then I get 1,000. Are you seeing? Yes. Now, remember I have a total of 2,000. So 2,000 minus the 1,000 for these two. Yes, I'm going to end up with 1,000. This 1,000 is for these two now, for adenine and thymine. But because adenine and thymine, when you combine them together, it is 1,000. So you divide by 2 and I get 500. It remains that this is 500, this is 500. I think I'm clear there. I think calculations in life science, those are some of the calculations. Another calculation could be amino acids. Amino acids. Yes. Amino acid, the last calculation. The last calculation, yes. Are we required to add a formula? Which formula? Where exactly? Do you mean on the percentage increase, percentage decrease? If you mean the percentage increase, percentage decrease, you have to apply the formula. Yeah, final minus initial divided by initial times 100. Yes. Then, don't forget that you have code, you have codon, you have anti-codon. Yes, you have the amino acid. Once I finish this, then I go to the rapid questions. Yes, the other questions which can come, but in most cases this will take, in most cases it takes, we don't deduct marks here, we always bring the question in the full. Okay. So here, code, what is a code? Code is from DNA. Codon is from messenger RNA. Yes. Anticodon is from transfer RNA. Amino acids forms a protein. Yes. So when does it become a protein? 50 amino acids and above is a protein. Less than that is a polypeptide chain. So how many codons form anticodons? One codon forms one anticodon, and one anticodon forms one amino acid. Am I clear? One cod forms one anticodon, one codon forms one anticodon forms one amino acid. So it's 1, 1, 1, 1. So when it comes to bases, how many bases? 3 bases forms one codon, one code. 3 bases form one codon. 3 bases forms one anticodon. 3 bases form one amino acid. Now, question can come. How many code if I have 20 codes? Yes? How many amino acids? Amino acids are there? Because 1 versus 1, then I'll have 20. How many bases? How many bases will be there? Because one code calls for 3 bases, is coded by 3 bases. So I'm going to say 20 times 3. So it gives me 60. Yeah. Oh, we can ask it in the reverse. Ne? I have 20 amino acids. I have 20 amino acids. Yes? How many bases are going to be there? Still is going to be 20 times. Yes. Or I have 20 bases. Okay, 21 bases. How many codes, anticodons, or amino acids are going to be there? Whatever, because they are 3. So it's going to be 21 divided by 3. So I am able to get how many are there. Yes. I think you are not lost. Yeah, these are some of simple questions they can bring and the people go off. There are some questions I'm not going to waste the time, like describe transcription, describe translation. Those ones you know. But these are the technicals which makes you to lose a distinction. This is where the marks go. This is where the distinction come from. If you use this distinction material, this book for the distinction material, yes, you get a distinction. But there are some few things which make you to lose that distinction because you did not interpret correctly. I think I'm clear there. Yes. Let me go to now the questions. Questions. Yes. Let me go to the questions and then I see where to get the answers. All right. DNA. I think I was clear about DNA yesterday. I think I tried to exhaust everything about DNA. I think let me just talk about meiosis, and then after that reproduction, and then if I get the remaining time, then I can talk about DNA. Yes. All right. Let me start with the meiosis. Okay, let me just see here because I just downloaded some few papers, just papers for, I didn't get them anywhere. Yeah, this is 2019 November question. Yes, it's 2019 November question. Look here. I'm just looking a question for DNA. Just looking for a question for DNA. Ne? Yeah. So, this is paper 1. Ne? This paper 1. DNA is supposed to be in paper 2. Yeah. Here is before. Yes. After that's a question of DNA. It's a question of DNA. It's another question of DNA. You understand? Yes. Okay. Let me just do one question and then we see. What is W? W is I'm waiting for the answer. Okay, if you look at W, this is, if this is a chain of W, it is polypeptide chain, not protein. I said that 50 and above amino acid, it forms a protein. If it is short like this, it can't be a protein. It's a polypeptide chain. What is X? Is amino acid. What is Y? It is transfer RNA. That's what I said yesterday, that in most cases when I bring this transfer RNA, messenger RNA, they ask you, what is this? What is this? What is Z? Z is anticodon. And then once you see a ribosome, it means that that is translation. Group molecule Y. What is molecule Y? We said is transfer RNA. What group of nitrogenous bases? Z. group of is this one, anticodon. We have talked about it. You see, there are three of them. Three of them, they make what we call, this is what I say, that three of them, they make what you call the anticodon. Three of them, three bases make anticodon. So this is what they are saying here. This is what they are saying here. 1, 2, 3. So the group, it forms the anticodon. Nitrogenous base 1, 1, 2, 3. We have seen it. Yes. DNA triplet, that code for X. That code for X. Where is X? X is here. And then this is Z. Because this is ACC. So it means that it's going to be A goes with the T. And then C goes with the G, and C goes with the G. So it's going to be TGG. Okay, Sunny. Sir, I would like to ask you, how does gene mutation occur during replication, and what are the effects? Okay. Then they're saying that, okay, Sunny, I'm coming to you. Let me finish this one and then I'm coming to you. They're saying that DNA triplet, we have seen it. Describe the process of transcription. Guys, I'm not going to waste time here. You cannot go to the paper without knowing DNA replication, transcription, translation. Three of them, yes. So here, every different color means a marking point. Every different color means a marking point. So make sure that use this distinction material. Yes. Then you are asking, how does the mutation, the gene mutation occur during DNA replication? The gene mutation, remember, when you talk about gene mutation, it's like a point mutation. It occurs at a specific point. For example, this part might be removed. This part might be removed, or one base is being replaced, and then what happens? Obviously, is going to be a different person. For example, I'm just saying, if you are Nkensani, and then I say you are Nkonsani, you are not the same person. You have changed. When I say Nkonsani, when you are Nkensani, I'm just using you an example. Yes, it will be is different. So when one base is replaced, it means something different. So this is what I'm saying. This is what I'm saying. Told you that the distinction material will answer all your questions you want. And they're saying that you have two alternatives. If a base changes in the sequence, and then it leads to the formation of new protein, then it means that the protein, the functioning will change. If the base codes for the same amino acid, if the base changes and then the amino acid changes, then it means that it will cause the protein to change, or the functioning of the protein. But if it changes and it changes, the base changes and does not change the amino acid, it means that yes, however much the protein, the mutation has occurred, but this mutation has no effect on the structure of the what? Of the protein. Let me show you what I'm saying. This is what I'm saying. What will happen if a mutation changes the base triplet 1, where is triplet 1, from ATG to ATA. So I need to know what is this. This is the codon. So if this is codon, it means that if this is codon, so I have to change this into codons. So this is going to be ATG is going to be, it's going to be, remember codon is found on amino acid. On messenger RNA. A goes with U. T goes with A, goes with the C. So it means that I have to look for this. Is UAC. UAC is trypsin. Is this one. Now it has changed it to ATA. ATA. A goes with U. T goes with A goes with U. So it is UAU, which is still the same thing. You see? Tracing. It means that however much the base changed, however much there is a change in the sequence of the DNA during DNA replication, You understand? However much, not DNA replication, during protein synthesis, yeah, it can occur even in DNA replication. Yes. However much has happened, but it codes for the same amino acid. Therefore, it is the same thing. So the protein will not change. Gensani, I can call you Nkensani. I can call you Mutwakai. Isn't it? So when I call Mutwakai, you are the same person. When I call you Nkensani, you are the same person. You will say yes, sir. You say yes, sir. So it means that you are the same person. It's the same amino acid. However much there is a change in the sequence of the bases. So because it has changed at a point of DNA, then we can call it point mutation. Or because the gene has changed, then we can call it gene mutation. I think I'm clear there. I'm in class. Then, uh, what happens if the the amino acid changes? So it means that sequence in the DNA will change the sequence in the messenger RNA, which will change the amino the sequence in amino acid. Yes? And then the protein will also change. But if the sequence in DNA changes the sequence in messenger RNA, and this codes for the same amino acid, then it means that the protein will not change. It will remain the same protein. So it will have the same functionalities. Yes. If you have A goes with, T goes with the and then U. Then you have A, So AAU means what? AAU is valine. And then AAA means what? AA is proline. You see now, it has changed. It's only A which changed here, but it changed from valine to proline. You understand? You understand? So basically, now it means that a different protein will be changed. Saying that I was asking in the DNA, the effect will remain the same. The effect will remain the same. It will bring about gene mutation. So whether it happens in transcription or it happens in DNA replication, the effect remains the same because that will code for a different amino acid, or when you're explaining, still explain it in the same way. But in this case, you explain it in starting from DNA replication. So in Kensani, it is the same. It is the same because it is the DNA which determines what is going to happen in transcription. I think I'm clear there. Any question? If you have any question, or yes, I will open the line so that before I end, I open the line so that if you have anything to say to people outside there, yes, so that be prepared soon as long as you are talking about or any advice you can give. Yes, if you have any advice, let me know in the comment. Then I'll open the line for you, and then you call, and then so that yes, meiosis. Meiosis questions here, we talked about a lot. Where does it occur? You know that we said that in animals, ovary to produce ova, testis to produce spermatozoa in male, plants, ovary to produce ovule, anthers to produce pollen grains. That one confuses students. Number 2, the function of meiosis. What is the function of meiosis? Is to produce haploid gametes. Number 2 is to produce haploid gametes. Number 2 is to produce, is to solve the doubling effect of chromosomes. So it solves that problem of the doubling effect. Number 3, it brings about variation. Variation. You resemble the way you resemble because of the meiosis which took place is one of the cause during the crossing over process. Then chromosomes, please don't be confused with the unreplicated chromosomes and the replicated chromosomes. Yes, I think I'm clear there. Number of chromosomes, we have talked about it. Haploid means one set of chromosomes. Diploid means two sets of chromosomes. Karyotype, we talked about the karyotype. Karyotype, it means that you are describing the type of nucleus. If you have anything here, let me know. Ne? I just want to rush to where distinction is being lost. You understand? Where distinctions is being lost. Process, you have to know. You remember them by IPMAT. Yes. And then crossing over. You can't go to the paper without knowing the process of crossing over. Yes. Chromosome, the homologous chromosome, they align together. Chromatids, they cross over at a point called the chiasma. And don't forget that it takes place in prophase 1. Then the genetic material is exchanged between the homologous chromatids, and then you form two chromosomes which are having the combination of genes. Yes. So the process here, you need to know. When you look at this, you have to know which phase is this. Please, you don't go to the paper without knowing this. Should I repeat it? You should not go to the paper, any paper, June, final, what, without knowing this. Yes, I'm rushing now so that I cover even the last part. But at least now you have 70% now, at least. Yeah, because this paper for term 1 is always a small paper, 60 marks. And then if they do it too much, 100 marks. And the more marks will go to the experimental questions. That's why I spend more time on the experimental questions. Yes, here they just do sampling in few questions, but I'll try to guide you still. And then you write your paper, any day you write it, still you'll pass it with the distinction as long as you follow what I'm saying. And you have to know, when there are two chromosomes, they are under meiosis 1. When it is single, you see, single, it is meiosis 2. You see, it's single. This one is alone, this one is alone. Yes, is meiosis 2. So now, if they are in the middle, it is the metaphase. If they are apart, you see, they are apart is anaphase. So because they are two, is meiosis 1. So it's going to be, because they are apart, it is anaphase. Therefore, it is anaphase 1. That's how you can identify. You understand? Yes. Importance of meiosis, I've talked about it. Causes of variation. Guys, when you talk about causes of variation, please take note. If they talk about meiosis, the causes of variation on meiosis, you only talk about crossing over, random arrangement of chromosomes during metaphase. Don't say random arrangement of chromosome. Wrong. Where? Random arrangement of chromosome during metaphase. Random fertilization, random mating. Mutation, yes, could be, but sometimes may not. It could even happen before meiosis. So, random arrangement, if they ask 3, 1, 2, you can add mutation. But if they ask 2 or 1, please start with this. Random fertilization and random mating, that is not under meiosis. It is under another phase. I think I'm clear there. Yes. Non-disjunction, failure of the chromosome to separate during metaphase. No, during anaphase 1 or anaphase 2. So failure of the chromosomes to separate during anaphase. So it is, you see now they are being pulled to one side. So it means that it's going to leave one cell with a short of chromosomes. I did all these classes, but now we are just trying to be fast. Ne? Yes. Tekofato, I think you are in a class. I'm not seeing your questions here. Tekofato, I'm not seeing your questions here. Can I see you? Yes. Another difference between meiosis 1 and meiosis 2. Here, please look at the phase. Phase 1, prophase. In meiosis 1, there is crossing over. Meiosis 2, there's no crossing over. You go to metaphase. Metaphase, chromosomes align along the equator randomly. You understand? In homologous pair. What is the cause of mutation? What is the cause of mutation? There are so many causes of mutation, but we don't ask you that. Yeah, it can be X-rays, it could be even ultraviolet rays, but we don't ask you. I only focus on the ATP, examination guideline. Let me say like that, examination guideline. Guys, don't waste time on the things which are not being asked. Thank you for maybe you didn't know, but if you are not getting it, follow the examination guideline or get the distinction material booklet, and then you'll be able to get all the marks. Yes. So here we are saying that you go to anaphase chromosomes separate, yes, to opposite poles. Homologous chromosomes. Yes. The other side is chromatids. Chromatids are the one which are separating. Are you seeing? Then mitosis, meiosis is the same thing. Mitosis, the way you see meiosis 1, it looks like meiosis. It looks like mitosis. No, meiosis 2. So here, somatic is found in the body cells, here in the reproductive cells. We don't have meiosis anywhere else. Genetically identical, here it is genetically non-identical. That's why you don't resemble exactly the same as your what? As your brothers. Even the so-called identical twins, they are not identical. If you're saying that they're identical, they must get the same marks in the class. Okay, they are identical. Yes. They are not identical. No one is identical to each other. However, because they look alike, we can say that they're identical. Yes. Yes. Let me go to the questions. Now, I was just perusing through. Yes, perusing through. I saw a question of, here's a question. Yes. Guys, if they don't say that this is a human cell, don't talk about the 46 chromosomes. Please don't waste the time. Don't talk about 46 chromosomes. Yes. So here, we are saying that, you see, it is the non-disjunction occurred here, you see. Non-disjunction occurred. They're saying that, let me just answer this question, as maybe when I'm answering them, I'll be able to get more questions. Identify what is A. A, it is Z. First answer all of them. Centrioles. Centrioles, they give what called the spindle fibers. Guys, if it is not under prophase or interphase, the first layer can't be a nuclear. Please don't say this is a nuclear membrane. It is cell membrane. This is a cell membrane. This is a nuclear membrane. The inside one is the nuclear. If you see one, it is a cell membrane. If you see there are two layers, the outside one is cell membrane, plasmalemma, whatever, depending on how you memorized it. The second one is a nuclear membrane. Yes. Are the phases of mitosis similar to those of meiosis? The phases of meiosis, the phases are the same. Prophase, metaphase, anaphase, telophase. But meiosis has prophase 1, metaphase 1, anaphase 1, telophase 1. And then also, prophase 2, metaphase 2, anaphase 2, telophase 2. But mitosis doesn't have that. Only has prophase, metaphase, anaphase, telophase. That's all. Yes. Then they're saying that, what is the B? B is a chromosome. What is C? C is a cell membrane. And then, is there anything else? No. What is happening here? This is cytokinesis, division of the nucleus. They're saying that name, give the number and the name of the phase which show the following: random arrangement of chromosomes at the equator. You see, that's why I told you that you have to know these phases. You have to know these phases, that when it is like this, it means this. Random arrangement of chromosomes during at the equator. Random arrangement of chromosomes at the equator. It means that they are in the middle. When they are in the middle, what does it mean? It means that it's metaphase. So when Don't look at the two cells. Look at the cell individually. Ne? Yes, look at it individually. So, if you look at this, yes, if you look at this, ne? This is the middle. Also, this is the middle. They're in the middle. However much you see that there are two cells, going through the same phase. Yes. You see how you can lose marks. You can even fail to find it. Yes. So the answer, they're saying phase, give the chromosome at the equator. Yeah. So it's going to be, give the number and the name of the phase which shows the chromosome. So the number is number 2. But which phase is it? Is it metaphase 1? You see chromosomes are single. So it's meiosis 1. It's meiosis 2. So it means that it's going to be metaphase 2. Metaphase 2. And then crossing over. Crossing over only takes place in prophase 1. Prophase 1. You see? So, Miche is asking, yes, prophase 1 occurs only in what? Yes. So now what does it mean? What does it mean, Dimple? Is in interphase part of meiosis? Interphase is not part of meiosis. No, it is not part of meiosis. It is just a phase, is a growth phase. You understand? So when you are explaining the phases, you are talking about prophase. We start with prophase. That's why we say that we ask you a question there, that when does DNA replication occur? It occurs during interphase or before the cell divides. It's the same thing. When there's the cell, before the cell divides, it means that the cell is under interphase. Yes. So interphase is not part of meiosis. Still is not part of mitosis. They're saying that now crossing over only takes place in prophase 1. So which one is showing crossing over? No. It is here. You see they are crossing over. So the answer is 4 and then prophase 1. When they ask you crossing over, you should not think on anything else. Think about, you must think about prophase 1. Non-disjunction, failure of the chromosomes to separate during anaphase. So now you know the what? The phase. So it is anaphase. Anaphase. But we need to know if the chromosomes are in pairs, therefore it's going to be anaphase 1. If they're single, it's going to be anaphase 2. So if you look at it here, yes, this is with this, you see? So the chromosomes are still in what? In pairs. So it means that this is the anaphase 1. So it's anaphase 1. How many chromosomes will be found? Guys, they didn't say it's a human cell. I'm saying it in your ears. They didn't say this is a human cell. Okay, how many chromosomes will be found? The cell at the end of meiosis shown in the diagram. Shown in the diagrams. How many? If you look at them, check. You see they are how many? They are 4. So it means that at the end of meiosis, so it's going to reduce by half. So it's going to be 2. You see, they're saying normal human sperm. Normal human. A sperm is always haploid. I talked about it here in the booklet at the beginning of meiosis. I say that sperms are always haploid. Yes, I said it here. You see, haploid, sperm, haploid, half of the chromosome number. Therefore, if they're saying that is a human cell, therefore, now you talk about the 46, the 23. Yes, so because the sperm is going to be haploid, therefore, the answer is going to be 23. Yes. Somatic cell of a normal. Somatic cell. Now when you talk about somatic cell, you're talking about the body. You're talking about the body. So the body is having 46. That's what you know. Of a mother who has a son with Down syndrome. It means that who has a son with Down syndrome. So it means that how can I identify the phase in the human cell? How can I identify a phase in a human cell? Okay, I'll come back to you. This is it, but they're saying Down syndrome. Remember Down syndrome, chromosome number 21 is extra, so plus 1. So it means that they're going to be 47. They're saying somatic cell. They're not saying the sperm. They're not saying the ovum. Nope. Here they say the sperm. Here they saying somatic cell, the body cell. I think I'm clear there. Yes. So the answer is going to be 47. I think I'm clear there. He's saying, how do I identify the phase? I say that you have to identify the phase using the process of IPMAT. The way I said it here, the IPMAT, which means that IPMAT, how do I identify the phase? Okay, let me just show you here. I say that IPMAT, I say that IPMAT, which means prophase, metaphase, anaphase, telophase. It can say preparation, middle, apart, and then terminal. Yes. So it means that preparation, middle, apart, terminal. You understand? Yes. So now, what is in preparation? If it is under crossing over, you just know that is the prophase. When it is under, it is in the middle. You see, it is in the middle. Yes. The chromosomes are in the middle. I told you that these things must be on the sides, on your sides. Yes. If it is on the sides, you have to turn the paper. So it is in the middle. So it is a meta phase. But if it is single, look, I can have this. I can have this. If it is single, you see, single, then it's going to be metaphase 2. If it is double, it's going to be metaphase 1. I think I'm clear there. And then if it is anaphase, ana means apart, you will see that they are moving to opposite poles. You see, they are moving to opposite poles. But because their chromat is moving, then it means that they are what? It is anaphase 2. So if it is the whole chromosome moving, then it is anaphase 1. So that's how you see them. And then in telophase, you can also see it using cytokinesis. Yeah, I think I have 15 minutes to finish. I think I'm done with that. Reproduction in vertebrates. I think even if I don't waste too much time in reproduction in vertebrates, this external fertilization, internal fertilization, viviparous, oviparous, and then okay, oviparous. It means that there is development is outside. The oviparous, it means that ovoviviparous development is inside the mother's body, but the baby or the embryo is obtaining nutrient from the yolk. Viviparous, the development is inside, and the baby is obtaining, or the embryo or the fetus is obtaining nutrients from the mother through the placenta. Yes. Fertilization is when the haploid nucleus of the sperm fuses with the haploid nucleus of the ovum to form a diploid zygote. If it happens outside, then it is external fertilization. If it happens inside the mother's body, it is internal fertilization. When you are born prepared, when you are born, the eyes are open. So this is what I'm saying, external fertilization. You see the sperms are happening outside. And if it is happening outside, definitely is oviparous. So internal fertilization, but still it is oviparous because the development is outside the mother's body. Yes, all this is oviparous. And then this is viviparous. The nutrients through the placenta. You see, the nutrients are being obtained through this placenta. Yes. And then here, it is obtained through the egg yolk. Is inside. You understand? Okay. There is something which I wanted to talk about here. Yes. All right. Okay. Then I was talking about precocial atricial. Yes. Precocial, you're born prepared. You can walk, you can move, you can defend yourself. Yes, you see, is born prepared. That's why it's called precocial. Young are able, they are mature, they're able to walk, they can able to defend themselves. They have down feathers. That's what it means. Then precocial, eyes are closed. They are helpless. They are what? They are helpless. Alright, too. Saying I should take my time. I'm not taking your time. All right. They are helpless. You see? So it means that you cannot go to the paper with we either bring precaution or attrition. We like them. We like them. If we don't bring them, we're gonna bring them in objectives because you can't miss it. Because this paper is fewer marks, is not 150 and is combining everything. We can have 2, 3, 4 questions from there. Yes. All right. Let me see here if I have a question concerning about it. It's supposed to be paper 1. It's supposed to be paper 1. I hope I have one. If I don't have one, then we just move to another question. I see, I don't see any question here. But if you go here, you'll find them there. Finger-like projection, division of the cytoplasm. Division of the cytoplasm is cytokinesis. We talked about it. Finger-like projection, extraembryonic membrane is chorionic villi. They're saying that structure of female reproductive system where semen are deposited, vagina, permanent scale. We don't talk about that. Hormone that stimulates the mammary glands. We don't talk about it, not yet there. But it's prolactin. They're saying period of development of the fetus. This is the gestation period. Use gestation period than pregnancy, if just in case. Structure of female reproductive system where fertilization takes place. Fallopian tube. Yes. Okay. I think they brought the question. Yeah, they brought it here. Yeah, I'm seeing here. Define what is oviparous. See? Yeah, such questions, please don't lose. I've seen here a question here. Yeah, this one. And also another question is for reproduction. Guys, I know a female reproductive system is easy. Yeah, it's easy. But we like to ask the male reproductive system more than the female reproductive system. I don't know why. Yes. Yeah. And you cannot be a girl and you fail this structure. Never. Why? Why do you fail it? Don't forget to stay virgin. But I know that you can't fail this structure. And you can't be a boy and fail your own structure. Why do you fail? You don't know yourself, you know others. I call it being insane. You know others and you don't know yourself. No. I know that you girls, you can't fail this structure. You know it better than boys. Yeah, that's what I know. Reason why, you must tell me. I don't know. But I know that you love it more than boys. Okay, what is one? Let's just label it. Yeah, it's not about labeling everything, but you can label it. You never know. We might have such a question in your paper. One is seminovesicle. Look, it starts from here. The first gland is called the seminovesicle. You see, the seminovesicle. The second gland is the prostate gland. The third gland is corpus gland. Then you move to urethra. Okay, number 1 is seminovesicle. Number 2 is the prostate gland. Number 3 is the vas deferens. Number 4 is epididymis. There is this boy who like to say epididymis. They will bring it and you fail it. Hey. Make sure that you have correct spelling. Yes, make sure. Ali, are you in class? I want to know. Make sure that you have correct spellings. Mpo, dimple. All right. 6 is the scrotum. Why do testes hang outside? Yeah, you must answer that. We say that to provide favorable temperature for sperm production, optimum temperature for sperm production. Sperms are produced 2 to 3 degrees less than the body temperature, which is 37.5. Yes. So meaning that the sperms are being produced around 35 degrees C. Scrotum protects. Testes produce testosterone and also sperms. Epididymis stores sperms temporarily. Vas deferens transports sperms from epididymis to the ejaculatory duct or urethra. Then seminal vesicle produce the alkaline fluid which neutralizes the acidity of the vagina. This one, Cowpass gland, produces the lubricant. Yes, lubricates. And then seminal vesicles produces nutrients. Produces what? Nutrients. I think I'm clear there. Yes. Structure of a sperm, we have done this several times. Acrosome, nucleus, uh, uh, middle piece, flagellum or tail, jelly-like layer, cytoplasm, nucleus. Is it clear? Yes. Name the process which involved which did the formation of the ovum. Formation of ovum is being done by oogenesis. Yes, they're saying involving meiosis is oogenesis. Remember, we have gamerogenesis, which means formation of gametes. Spermatogenesis, formation of the spermatozoa, and oogenesis, formation of the what? The ova. So here they're talking about the ovum. Write down the letter of the part that is of the part of the sperm that enter the ovum. The letter of the part which enters the ovum. Remember, this tail remains out, and the head contains the enzyme which digests the membrane of the ovum during fertilization. So the only part which enters is the nucleus, which is D. Write down the letter of two parts that enables the sperm to move towards the ovum. That is the middle piece, which contains the mitochondria to provide the energy so that the sperm can move forward, and also the tail, long tail, which provide the propulsion force for the sperm to move forward. I think I'm also clear there. The brain, you have not yet reached there. Still, this is the meiosis. You see? So I think, guys, you're gonna smash this paper. Yeah, because there is no new question they're gonna set you. Any day you write it, whether I'm sorry for those who already wrote their papers in different provinces. Yes, but good luck for other papers. Yes. All right. They're saying that what is A is a centromere. You see, centromere. When it is under crossing over is a chiasma. You understand? Yes. Centromere. Two chromosomes under this, we call it bivalent. They are homologous chromosomes, but it's called bivalent. 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 Yeah. I think I'm clear there. Yes. Then they are saying, where am I? Okay. What is C? C is spindle fibers. And then what is okay, this is a bivalent, ne? Yeah, it is not bivalent. Bivalent is supposed to be under crossing over, ne? Yes, this is not crossing over. You see, this is what I say, that these things are supposed to be inside. So now you don't know which phase is this. It is meta phase, so you have to change your paper like this so that it faces opposite. You see that now they're in the middle. You see, they're in the middle. Yes, they're in the middle. So it means that you're able to answer it. Yeah, so you have to tilt your paper until you see that those things that are in the what? They're in the middle. Yes, I only have very few minutes now, 2 minutes. If there is anybody who wants to talk something, please let me know. This is the menstrual cycle. Based on what we said, in most cases, we leave 2 to 3 weeks, 2 weeks before so the menstrual cycle must be involved. You have to know the hormones which involved in the menstrual cycle. I cannot repeat Mr. Who? Fallope method now. Why? Because the time is not enough. What is the reason behind boy having to go through circumcision? When? Minengi. Why do you, boys, why should you be circumcised? Are you circumcised, boys? Yeah, basically, circumcision is about protection you from some diseases, STI, like HIV. You have 65% safe if you are circumcised not to get HIV. You understand? And other sexually transmitted diseases. Yeah, it means that the let me say the machine is hard, ne? So friction may not be enough to create some wounds, ne? Some wounds on the machine. When I say machine, I think you understand. Unless if you have a small machine, okay, okay, a machine, ne? Yes. So now it means that blood exchange might be minimized. You understand? Yes, that's why you're supposed to be circumcised. Also, to be clean. To be clean. Yes. So, guys, menstrual cycle, I'll only say, please don't go without knowing the hormones which are involved in the menstrual cycle. FSH, LH, estrogen, and then progesterone. Progesterone. Let me write it slowly. Progesterone. Yes, these two are from the ovarian cycle. They are from the ovary. They don't work under ovarian cycle. They work under uterine cycle. These two, they are from the brain. I think you are clear there. Stimulates the development of the Graafian follicle to produce ovum. This one triggers ovulation, is very high on day 14. Ne? You must know that. Very high on day 14. Triggers ovulation, also changes the empty Graafian follicle to corpus luteum. This one changes the, we say that it prepares the uterus for implantation. And then lastly, progesterone maintains pregnancy. It maintains pregnancy. How? By making the endometrium wall more thicker, more vascular, and more granular. That word more must be there. Development of the zygote until implantation. I think, yeah, I've not gone through here, but I think it is clear there. Please don't go without knowing those. Development of the zygote until implantation. I'm talking about this is the copulation is here. Yes, development of the zygote until implantation. Yeah, is here. Yeah, you can also do that, but it is out of the 2 weeks. So I don't know whether they're gonna bring it or they won't bring it. But if they bring it, please don't forget that this is fertilization. This is a zygote. It undergoes mitosis to form a ball of cells, which undergoes mitosis to form a hollow ball of cells, and then you tell them morula, blastula, and then forms the outer layer, which you call the chorion. The chorion forms finger-like projection, which forms chorionic villi, and then they are used for implantation. I think that's it. Ali, I've not seen you in my class. Is there anyone who has a question, who wants to call? If you want to call or tell some people, you can call on WhatsApp. Then they will be able to hear you properly on live. Yes, you can call me on my WhatsApp. Is 0848331738. Yes. I don't know. Yes, I said 0848331738. Yes, that's my number. Always. Thank you, sir, for the explanation, guys. Charlotte, yes, all right. Guys, if you have any question, those who are writing their papers, because March is that's when you write the paper, make sure that you go through what I said. I wish you the best, the best, the best. Hey, someone was asking me, is sometimes caused by infrared? Yes, radiation. Yes, of course. Also, but the infrared radiations, they are not that strong. They're not that strong. Infrared radiations, these are the radiation used at home, TV, the remotes, those are the infrareds. Yes. Yeah, but the ultraviolet radiations are the one which are more stronger. Ne? Yes. Uh-huh. Nkensani. Nkensani. I think that is it. Someone is telling me that. All right. Oh, he's talking about the distinction material. Yes. Guys, I think I'm going to stop here. Don't forget. Maybe let me just show you something here. How do you access? How do you access? How do you access the book for distinction material? How do you access the those who want to call, I'm still online just for 2 minutes. Just go there and then say type Thunder a Duke. So it's gonna bring you this. When it brings you Thunder a Duke, yes. So you press where you want to press. Yes, it's gonna bring the first page, and this first page, this is how you can access all more resources. You go to grade 7 to 12. Once you click on it, grade 7 to 12, yes, grade 7 to 12, then it's gonna bring the different options. Yeah, there is past paper questions, there is study notes, there is study videos. Now when talk about the study videos, it means that you'll be able to get all the videos, the different questions. You just type the topic and say Thunder Educ. Yeah, like that. So when you go, for example, study notes, yes, study notes. When you go to the study notes, yes, is opening. Study notes, yes, it's gonna give you the study notes here. You see? So now you choose the subject you want. Ne? Grade 10, grade 12. So they give you life science. You see? So when you find there, you'll find there even questions. Ne? Past paper questions. And then we keep adding. Ne? Yes. Past paper questions. We're still adding. Yes. Watch the video. Study booklets. Yes. Test and memos. So that's how you can access it. Ne? Yes. So thank you very much. I salute you for those who have been with me from the beginning to the end. Don't forget to subscribe, like, and share to others for these daily new videos. Stay blessed.